at the University of Leeds and the White House and West Head groups. And we talk about the first chapter of my PhD thesis today on dysregulation of circRNA-miRNA networks during lytic reactivation of KSHV. So circular RNAs are expressed from genes that are capable of undergoing splicing, and they're formed via a process called back splicing, which is in contrast to canonical forward splicing, where you have a sort of downstream 3' splice site is spliced. Sorry, an upstream 3' splice site is spliced to a downstream 5' splice site in a kind of linear fashion to build up sort of beads on a string in a way of the exons. And that becomes the mRNA and it's exported, and partly that's linear as a 5' end and a 3' end. However, when with circRNAs, they form via this sort of reverse process called back splicing, which is where a downstream 3' splice site is spliced to an upstream 5' splice site, to result in the formation of an RNA circle, hence our circular RNA, with a unique backsplice sequence, which is where the sequences are kind of in reverse order or orientation to what you would see encoded genomically. And that's one way how we're able to quantify them. But the unique sort of shape and structure and properties of circRNAs confers various features towards them. For example, the fact that they're covalently closed circles and they're linked via a 5' to 3' linkage, not like a 2' to 3' linkage, 2' to 5' linkage that you get for RNA lariats or things that are produced as a side product of a regular splicing. It means they're really stable, they're resistant to exonucleases, and they're resistant to debranching enzyme and stuff like that. And because of this, they have quite high half-lives, and although their sort of biogenesis rates can be quite low, they can accumulate to quite high numbers just to the fact that they're retained within, because they're quite highly stable and they get retained within the cell for a long period of time. They're pretty ubiquitous and prevalent. They're expressed from over 1/3 of human genes, and they've been found in pretty much all eukaryotes studied so far as I know, even higher and lower eukaryotes. They've been shown to be differentially expressed in thousands of different conditions, including viral infection, in various diseases, in various bacterial infections, particularly in cancer and things like that. So there's some sort of circumstantial evidence linking them to a functional and a phenotypic role. So there's been many proposed methods to how they work, but the one I'm going to focus mostly on today is as miRNA sponges, and that's the one that seems to be the major focus at the moment with the most papers published on it. So the idea of a circRNA acting as a miRNA sponge is, if you imagine a circRNA is predicted to be able to bind a certain amount of miRNAs, and that's due to shared sequence complementarity between the circRNA and the miRNA. And in this way, the circRNA is able to sequester away a set number of miRNA from a free pool of available miRNAs, that so reducing the effective amount or pool of miRNAs that's able to then go on and repress transcripts. So by modulating the abundance of the circRNA, it's presumed you can modulate the abundance of the miRNA and thus modulate the abundance of mRNA transcripts that those miRNAs are able to repress. Thus, a circRNA could potentially impact quite a broad range of transcripts, impact quite a lot of their expression by changing their abundances. And it's thought that this could have some control over distinct pathways or processes in quite a kind of almost a specific but broad manner. It's a bit more complicated than that diagram, because it's not as simple as like a kind of hierarchical tree structure. It's much more of a network, because you can have circRNAs that bind to the same miRNAs, they bind to different miRNAs, and the miRNAs affect different transcripts. So it's quite a dynamic and complicated system. But one main thing I want to get through during this presentation is kind of, you expect there to be a sort of a reciprocal relationship between circRNAs, miRNAs, and the mRNAs that the miRNAs target. Because if the circRNA were to go up, you'd expect it to bind more miRNA, that sequester more out from the global pool, and thus result in a derepression of transcripts, and thus an upregulation of the transcripts that miRNA targets. And obviously the same is true vice versa. If the circRNA goes down, you expect the opposite to happen. What we think is happening is, sometime during lytic reactivation, KSHV triggers the dysregulation of certain circRNAs or sets of circRNAs. I'm not quite sure at the moment, but we think this may have pro or antiviral roles, and that it's able to dysregulate miRNA networks and thus the mRNAs themselves. And it could be in quite a specific targeted way, or it could be a bit broad. But as I say, we're not too sure if it's a kind of host response to the virus in an antiviral nature, because circRNAs have been shown to have quite strong innate antiviral roles, or whether it's the virus's specifically, it's bringing about this dysregulation to promote its own replication. So KSHV itself, or Kaposi Sarcoma-associated Herpesvirus, is a large double-stranded DNA oncogenic gamma herpes virus. It's the cause of several cancers such as Kaposi sarcoma, primary fusion lymphoma, and also something like things like multicentric Castleman's disease. Its KS in particular is very prevalent in AIDS-associated areas where AIDS is still prevalent, so places like sub-Saharan Africa, where there's still a large issue with AIDS there. And I believe it's the second most prevalent cancer, or at least AIDS-associated prevalent cancer in regions like that. So being a herpes virus, it exhibits a kind of biphasic life cycle where it will generally exist as in a latent state. It has minimal gene expression and minimal viral replication. It's just kind of maintained effectively as an episome in the nucleus, where it's not doing a huge amount, but expressing some miRNAs and some various proteins that aim to, just prevent it from being lost, not necessarily prevent it and allow it to replicate rapidly. However, what you get is you get something, a lytic trigger will come along, and there's various things that can do that, like reactive oxygen species or co-infection with something else can trigger the expression of of a gene called RTA, which is a virally encoded gene, which goes on and binds to viral and host promoters, thus increasing the expression and upregulated gene expression of both the viral genes, then also modulating host gene expression. And when it comes to the viral genes, it kind of exhibits this sort of cascade effect. It's called the lytic cascade, and it triggers this kind of, it's what triggers lytic replication and is lytic replication. But you start off with the immediate early genes that will be upregulated and expressed, then you get the early genes, then you get the late genes. And the late genes are things like structural proteins and things that help build more new virions. Because throughout all this time, you have increasing gene expression from the virus. It massively ramps up its gene expression, but it also starts actually productively replicating, not just maintaining itself within the cells. So I didn't generate any of the data. I'm a bioinformatician. I'm just working with the data that I was given, but this is the general sort of setup for the data when I joined. So we work in BCBL1 cells, which is a primary fusion lymphoma derived cell line. It's latent, and it's a B-related cell line. It's latently infected with KSHV, but it also includes this RTA gene under the control of a DOX-inducible promoter. And the idea was at 0 hours before you've added any DOX, you take an RNA sample from that, and that's your latent sample because it's unreactivated and it's just undergoing latent expression. You then introduce DOX and wait 24 hours and take the RNA from that, and that becomes your lytic gene expression. That becomes your lytic sample, and you take that RNA forward. So for both of these time points, we generate circRNA-miRNA data, small RNA-seq data, then total RNA-seq data, did quantification, alignment, and then differential expression on these, got the miRNA-target predictions or interactions from various sources I'll go into a sec, constructed the circRNA network, which you see down there, and then performed fairly network stats and clustering and various analyses on them. So I'll just go through the transcriptomics. I don't have a huge amount of time to talk about these, and there's a bit more of a story here if you want to ask any questions, but just focusing on so we ended up with 130 differentially expressed circRNAs, with most of them being upregulated. We ended up with 21 differentially expressed miRNAs, with 16 being down, with a majority being downregulated. And we ended up with about 4,000 mRNAs or linear RNAs, with most of them generally being skewed to be upregulated. And this is interesting because it kind of mirrors that thing I showed before, where you imagine the circRNA would go up, miRNA would go down, and there's results in a derepression of the transcripts resulting in that upregulation. There's some interesting sort of circumstantial evidence there, linking it back to the model that we're trying to investigate, basically. So to build the network, as I said, we got miRNA-target interaction data. So for the miRNA and the miRNA-mRNA interactions, I used 5 databases. These were Encuri, NBINTER, MERTAR-based target scan, and MERDB. I retained interactions if they were experimentally validated and present in at least 3 out of 5 of the databases. However, because things like circRNAs are quite new, they're not well studied, and also because the KSHV transcripts aren't nearly well studied as human transcripts, I couldn't really rely on any databases for the interactions. So what I did is I did some simple miRNA-target predictions between the miRNAs and the target sequences. I used two fairly commonly used target algorithms, Peter and Miranda. And because each individual miRNA-target algorithm is it's got quite a high false positive rate, I just took the intersection between two and kept those as my interactions. As I said, I did this for the miRNA-circRNA interactions, for the miRNA KSHV UTR interactions, and the KSHV ORF interactions separately. So from this, I was able to construct my network, and you can see it's not that massive a network as far as biological networks go. It's got 50 miRNAs in it, again, most of them being upregulated, 20 circRNAs, 50 miRNAs, most of them being downregulated, 20 circRNAs, most of them being upregulated, 89 host transcripts, again, most of them upregulated, and 35 viral transcripts. All of them upregulated because the virus is going through its lytic cycle. It's starting to express its genes, and it's just basically everything is increased in expression. So you can start picking out some kind of structures in this network just by eye. You see these kind of almost like floret structures here, where you get, what it effectively is it's a miRNA and it's all the targets it's able to bind. But you also get some interesting things, like you see a lot of the circRNAs are relatively peripheral to the network. They're not too central to a lot of the nodes, and they're kind of more targets of the miRNAs than anything else. However, there is this kind of dense core in the middle that contains a lot of viral genes and also a few miRNAs and circRNAs. We'll focus a little bit on that later. But the first thing I wanted to have a look at is why specifically these miRNAs that I'm focused on, like, are they particularly important? And the idea behind, or the logic behind this, is that a lot of the studies that look into miRNA circRNA networks, and they'll identify miRNAs, circRNAs, and mRNAs as being differentially or co-differentially expressed. So they'll show that they change in a consistent way between two conditions. But then they do miRNA target prediction, and then they just assume that as a kind of causal relationship, that it is in fact this circRNA, this specific circRNA that's affecting that specific miRNA. The problem with that is you would expect a certain amount of miRNAs to bind to circRNAs, or anything really, just by chance, because miRNAs are quite short. They're only about 22 nucleotides, whereas circRNAs can be hundreds to thousands of nucleotides long, and there's quite a lot of them. So what I wanted to do was specifically show that my differentially expressed miRNAs specifically bound to my differentially expressed circRNAs relative to, a kind of like a background set of miRNAs that aren't differentially expressed, or at least aren't that special by some metric. So the approach I used for this was a fairly simplistic randomization approach. What I would do is I would just pick 21 miRNAs, the total number of miRNAs that were differentially expressed, at random from all the miRNAs in which differential expression was performed. So that was about a list of about 250, and it was basically the ones that were detectable above the thresholds, but not necessarily showed significant differential expression. So the next step was to perform miRNA target interactions between the miRNAs and this 21 randomly selected miRNAs and the differentially expressed circRNAs. I repeated that process 1,000 times to compute a null distribution, where it's just measuring the number of miRNA binding sites in the circRNAs. And the idea is that kind of represents sort of a null hypothesis, in the sense that, this is where there's no this would be if there's no significant enrichment of miRNAs, of my differentially expressed miRNAs in my circRNAs, if it's not significant, basically. And from this, I was able to compute a p-value, and that's just simply the proportion of subsets of the randomly selected miRNAs, in which the total number of miRNA binding sites of these randomly selected miRNAs and the circRNAs was greater than or equal to the total number of miRNA binding sites for the actual differentially expressed miRNAs, so the actual miRNAs that we observe. And when I did this, and I didn't just do it for the circRNAs, I did it for the KSHV, both the 3' UTRs and the open reading frames themselves. And for all three of these biotypes, we saw that, yes, there's a significant enrichment of the differentially expressed miRNAs in dark blue versus the randomly selected miRNAs in light blue. So we got p less than 0.005, 0.001 here for the UTRs. And for the ORFs, it's actually beyond the granularity of the simulation, so the significance could be a lot less. And on top of it being significant for just the intersection of miRNAs, it's also significant for the individual algorithms as well. So it's not like one algorithm is biasing this necessarily. So the next thing I wanted to look at is do some fairly simplistic network stats on it. So look at the network from the perspective of centrality metrics and things like that, in order to rank the nodes to know which ones to focus on, which ones were most important to the network for information flow and various properties. However, when I was starting off doing this, it it was difficult finding a sort of a robust theoretical framework to work with that would let me know what centrality metric would be best to use, because there's loads of them and it's quite hard to not be biased in how you apply it to your network. However, I did find this one method, Sinner, that worked really well. What this does is it will compute these, so up to 49 centrality measures on the nodes of the network to get to, and that will result in a data frame where you have the nodes along one axis and then the measures of centrality along another. So each element of the data frame or the matrix would contain a centrality measure for that particular node and for all the centrality measures, basically. And what you do is you run a principal components analysis on this data frame. You retain however many principal components describe 80% of the variance, and then you rank the centrality metrics based on the information retained, and this is in the form of contributions to principal components. And because you expect a certain amount of contribution just by chance, anything below that's what the red line's representing, basically. And we're interested in anything that's above the red line on this graph, effectively. So taking that data frame, I plotted it as a heat map, and I sorted it by just degree. And the idea of this is it's sorted physically by degree, but it's colored by that specific centrality metric. So subgraphs colored in order of the centrality metric, but it's actually physically ordered by degree. I won't focus too much on this diagram figure because I could probably talk about it for quite a while, but I'll just take you through some sort of basic things and the general kind of things that to take away from it. So the first thing to note is in the light blue represents miRNAs, and we see that they're dominating the centrality rankings for the degree, but then for a lot of the other ones as well. We have quite a few KSHV transcripts that rank quite highly. We have some circRNAs that rank quite highly, and then the miRNAs are generally a bit lower down. However, it's not quite so simple as you see. There's a bit of a mix there. However, some miRNAs in particular tend to rank really highly and consistently highly. That's the important thing across different centrality metrics. So these tend to be miR30B and 30C13P, and also 3689A3P. And these are quite interesting miRNAs because in some other analysis I've done, but I didn't get time to show, they're predicted to bind, they're predicted to bind a high proportion of viral transcripts relative to a lot of the other ones they're binding. They're what defines a lot of the viral miRNA links in the network. And also it's known that the miR30 family has important roles in KSHV biology, particularly in the biology of lytic reactivation. So it's an interesting link there. There's also, although there's 20 circRNAs in the network, as I say, most of them tend to be a bit middle-ish and not that highly ranking, or sort of central to the network, at least by some metrics. However, these 4 circRNAs here tend to rank fairly consistently highly. It's these circRNAs that we'll focus on mostly going forward. But as I was saying, the KSHV transcripts tend to rank quite highly, but you've got to remember that it's hard to compare those directly to the mRNA transcripts because of the differing sources of evidence. With the miRNA-target experimentally validated, the miRNA-mRNA interaction data is experimentally validated, which means it's going to be a lot more sparse than the miRNA-target, miRNA KSHV transcript interaction data, because that's just based on prediction alone, and that's much less sparse data. So what's quite interesting about these miRNAs and the circRNAs I focus on is they do sort of inhabit this little cluster here in the center. So the next thing I wanted to do is do some more formal clustering rather than just kind of picking it out by eye. And I didn't really see much in the way of a framework to go about clustering cirRNA networks. So I wanted to be a bit more stripped back and not overcomplicate things and be quite empirical about it, as I say, not really biasing using one specific method. And the way I went about doing this is use 4 relatively simple clustering methods. Edge between us, info map, walk track with 5 steps, and then fast and greedy. And these are all available with, I think, iGraph or various R programs. Let's say they're quite commonly used. But so this I ended up with 4 sets of modules for each for the network. I then computed a 4-way Jacquard similarity index between these methods, with the JSI just being a similarity measure between sets, or lists of sets rather. It goes from 0 to 1, with 1 being more similar and being 100% similar, and 0 being there's no common elements in either one. So using this JSI metric, I was then able to generate what I call meta modules, which are just modules that show a JSI greater than 0.5 in the network. And by using this JSI score, I was able to give me a bit of a sort of a metric to base on sort of robustness of the network to various techniques. That was the idea behind it anyway. So from the results of this is plotted in this network here with the clusters here, and you can see the coloring of the clusters represents the underlying GSI. Now, for most of these modules, A through D, they're not very interesting, right? It's just the miRNA and the targets it's predicted to bind, which we already know there's some association there. We can already sort of infer clustering just based on the knowledge of how we made the network. So they're not that interested in them themselves. However, what is interesting is this sort of central cluster does indeed come out as a cluster itself with a fairly high GSI of about 0.65 to 0.7. I say there's lots of kind of extraneous nodes that aren't necessarily included in the consensus in the meta module, but might be included in individual other ones. But these sort of this core network of about, I think it's about 25 nodes comes through quite strongly. Again, what's quite interesting about this is it's basically centered on these two miRNAs, miR30B and 3689A3P, to these two in the middle, which are the ones that bind to a lot of KSHV transcripts. And as we can see, there's quite a few KSHV transcripts in this module, more so than their actual host mRNAs, as well as 3 out of the 4 high-ranking circRNAs from the centrality analysis I did. So it's quite tempting to speculate with this this sort of cluster here that's playing a role in KSHV lytic reactivation, as the fact that we know the 30B and 3689A have been validated to show differential expression. However, unfortunately, the circRNAs haven't as of yet due to COVID and the restrictions that's placed on us. But we're hopeful that this cluster could be playing a role in the KSHV biology, because if you think about when the virus is going through its lytic reactivation, it wants to increase the expression of the mRNAs that it's producing, and by de-repressing them through down-regulation of miRNAs, that might be one method to do it. So just in summary, I'm going to go over what I was talking about and what we were able to find. So we want to investigate the dysregulation of circRNA-miRNA networks during lytic reactivation of KSHV. We identified distinct subsets of miRNAs, circRNAs, and mRNAs that are dysregulated, and we were able to link them up together via various target interaction methods. We were able to show that the dysregulated miRNAs specifically show a statistically significant enrichment in the dysregulated circRNAs, but also in KSHV UTRs and open reading frames. We showed a subset of the circRNAs and miRNAs rank highly in centrality analyses. Importantly, they rank consistently highly across the different methods. We also performed some clustering to reveal a sort of dense core containing two of these miRNAs and 3 of these circRNAs that could rank consistently highly, and also a high proportion of viral transcripts. So the last thing to do is just to thank my supervisors, David Westhead and Adrian Whitehouse, as well as Kate Sharp and Becky Foster, who are doing a lot of the wet lab validation of this, and then just the general Leeds Omics community for being very supportive during my PhD. So thanks, and also thanks for listening from you guys, and have you got any questions?
Thank you, Mr. Ewan McDonnell, for your wonderful talk.
Cheers, man.
Any question from audience, write in comment box.
So if you want to directly get in contact with me, actually, I've got a Twitter and an email. So just share that on the screen. If you have any questions you want to.
I guess there's no question. Okay. I would like to remind audience, if you want to know about the upcoming webinar or how you can present your research and be part of this initiative, you can follow us on Twitter. Another thing is that we arrange 2 webinars per week. So stay connected with us, and good day and take care. Thank you.